blood types were found in 1901 by a man named Dr. Karl Landsteiner. He found 4 blood types. Those blood types were A, B, AB, and O. Not a lot of information was known about blood types at the turn of this century. And what they were finding out was that when there were patients in need, and they would often transfuse blood as a last-ditch effort to save their life, many of the patients would wind up succumbing to that blood transfusion. They would die. Now, it was unknown why they would die. It would only assume that they were too far gone in order to be saved. But it turns out that the blood that was given to them could actually have been the cause that was killing these patients. So by identifying these blood types, we were able to find out that only certain blood types could be given to certain individuals. And if you gave them the wrong blood type, what would happen was that incoming blood would coagulate inside of their body, which is very dangerous when you plug up a blood vessel because it doesn't allow any nutrients or oxygen flow past that blocking point. Another way that blood typing was helpful was in forensic science in the early 1900s. By identifying a particular blood type that would allow you to perhaps rule out a suspect who didn't have the same blood type as was left on a victim or at a crime scene. So ultimately, we couldn't pinpoint identification like DNA does. It's going to take another 80 years for that to happen. But you have a beginning of using blood as a particular evidence type that could rule out some people as probable suspects at crime scenes. Dr. Landsteiner found that there were 3 alleles that controlled blood types. They were A, B, and O. But they led to 4 blood types. So we'll figure that out in a minute. But let's talk about the word allele for 1 second. Alleles were alternative forms of genes. When you study Mendelian genetics in your first biology experience, most of the time you were taught that there were 2 alleles per trait. So each characteristic was governed by 2 alleles. Sometimes you got the same one, sometimes they were different, and in both cases, it could often lead to different variants of that characteristics. It might lead to 2 different outcomes. For example, if we were talking about Mendel and his pea plants, you might have a short pea plant, you may have a tall pea plant, you may have purple flowers, you may have white flowers, etc. So, how are there 4 blood types with only 3 alleles? For the answer to this, we got to do a little bit of reviewing. Let's start here. So here we see a woman, and we see a small section of tissue that has been magnified many times so we can see a cluster of cells from within her body. If we then pinpoint in and zero in on one of those cells, we can see all the things that make up the cells. We have the cell membrane, we see the cytoplasm in between, and we have the nucleus. Now, within the nucleus, we can see these little rod-shaped structures right there. Those are chromosomes. And chromosomes are made of the nucleic acid DNA. So if we tried to unwind this, what we'd come up with is essentially a loose piece of DNA. And if we continue to unwind it, letting it release from all of these histone proteins like beads on a string, and we stretch it out, what we're going to get to is plain and simple DNA. And if we unwind the DNA and take a look inside, remember we have A's, T's, G's, and C's that make up segments of that DNA. Now, when you have a segment of DNA that codes for a particular protein, for example, let's say we have this little stretch right here, and what it does is it builds a protein from it. We call that a gene. So a gene is ultimately a stretch of DNA that codes for a working protein. Now, how does the term allele fit in there? Well, we can take a look at genes versus alleles to come up with some ideas. So genes are segments of DNA which control a specific trait, usually producing a protein, but sometimes regulatory. And an allele is a variant of a particular gene. So ultimately, we can have different characteristics that come from one gene. So normally, genes will determine a particular trait. But if you have variants of those genes, you may have variations of that trait. And us as a human species, remember that we get half of our DNA from mom and we get half of our DNA from dad. Let's take a look at the next diagram. These are what we call homologous chromosomes. So if you remember from your first biology class, humans have 23 pairs of chromosomes. We got one of them from mom and we got one of them from dad 23 times. So let's imagine that this chromosome here is from dad and this chromosome here is from mom. It's a pair of homologous chromosomes. One of them came from the egg and one of them came from the sperm to ultimately produce this individual with this chromosome pair. Now, along the DNA, along the chromosome, there are stretches that code for particular characteristics. And here you see that dad and mom both have the same code of capital letter A. We're going to say that when you have the exact same allele twice, that it's what we term homozygous. And we're going to imagine that every time you get a capital letter A, we're going to call that dominant, which means that the characteristic will be expressed when someone receives a capital A. So in this case, we receive two capital A's, which means 100% this particular trait is going to be expressed because of that combination. Now, in the second gene position, notice that father donated a lowercase b and the mom donated an uppercase b. So what that means, if we stay by the rules of dominance and recessiveness, that she contains the dominant allele and he has the recessive allele. Now, recessive alleles typically don't code for proteins, or if so, they do in very small amounts. But mom's dominant allele here will definitely code for ultimately a trait that will be expressed over the recessive gene. Now, in the third gene down, we have two lowercase c's, which means both mom and dad are recessive. So the recessive trait will then be expressed in what we call the phenotype, the overall characteristics of an organism. The last one on the top half of the chromosome is letter lowercase d, which both mom and dad donated to this individual. And we call that homozygous recessive. So the capital A's were homozygous dominant. The lowercase d's were homozygous recessive because it's the same letter for the allele twice. But that second trait we looked at, the B's, we had one capital letter and we had one lowercase letter. And we call that heterozygous. Okay, so homozygous is if they're the same. Heterozygous is if they're different. I think they forgot a little E here between the T and the R. Heterozygous. Now, on the second half of the gene, we can see big E, little e, that's heterozygous. Big F, that would be homozygous dominant. I'm going to skip this one for a second, but down here we see I and I, or L and L, and they're the same, so we're going to assume that in this case here that they're homozygous recessive. Now, in this case right here, normally we would have a big G and a little g, but what we have here is a G and an H. Now, the only way we can get a G and an H is if there's a third potential allele that has some influence on the overall trait of this organism. So if we were playing by the rules of Mendelian genetics, very simple Mendelian genetics, we'd have some form of G on both chromosomes for mom and dad. But now we have an H there, which means there's a third possibility. Now, blood types are going to utilize that third allele as well. So ultimately, any trait that's a consequence of 3 or more alleles is going to be called a polygenic trait. So with blood, we're going to see 4 different possible phenotypes based on 3 different alleles. Let's also review the idea of phenotype versus genotype. So genotype is essentially the allele combinations that code for some characteristics that we see in an organism's phenotype. The phenotype is what an organism expresses outwardly. So we can study the phenotypes, the overall characteristics of an organism, which are based on genotypes, or the allele combinations that produce those variances. So for example, for A blood, the genotype is going to be IA. That's one of the possibilities. But remember, we also have this heterozygous condition, which means that we could have a dominant allele and we could have a recessive allele. And that's what it would look like for the A type of blood. If we simplified it, we'd say that both mom and dad donated a capital A each, or mom and dad, it doesn't matter which one of them donated an A allele and one of them donated an O allele. For B-blooded individuals, the code for genotype is IB, IB, or IB lowercase I. To simplify that, mom and dad both donated a B allele, or one of them donated a B and the other one donated an O. For an AB blooded individual, IA from one parent and IB from the other parent. To simplify it, one parent donated an A, one parent donated a B. And for O blooded individuals, lowercase I, which means each parent donated an O. So you can see for both the A blood and the B blood, there's two possible combinations that allows someone to be characterized as having that type of blood. For AB blood, there's only one possible combination, which is each parent either donated A or B. And then for O blood, both parents donated the recessive allele, lowercase I, or to simplify it, donated an O allele. Let's take a look at this question here at the bottom for a second. It says, how does codominance play into blood types? Well, if you take a look at the genotypes for A blood and B blood, notice that in this case here, the dominant allele was donated twice, which is easy to understand that that would translate into a person having A blood. But then notice that in this case, the A allele got donated by one parent and the recessive allele that gets masked was also donated by the other parent. In this case, the person also has A blood. Similarly, if you look at the B-blooded person, in this case here, both parents donated the B allele, or the other possibility is one parent donated a B and the other parent donated the lowercase I allele, or the recessive allele, which gets masked. Now, in this case here, we definitely can see that there's a dominant and we have a recessive. But in the case of codominance, in the case of AB blood, notice that both of those dominant alleles get donated, and the overall characteristic or phenotype of the blood is AB blood. So they're both expressed in the blood itself, which we'll see in a minute in a diagram. And then our last case here, we don't have any dominant alleles. We just have the recessive alleles, little I, which translates into O blood for phenotype. So let's take a look at what's going on with these blood types. Okay, for this we'll do a little bit of drawing. So let's imagine we have a red blood cell. We already learned what those look like. Little biconcave discs. There's a little kind of hole in the center that is sealed up. But on the outside of the cell, this is where it's important when we talk about blood types, because these alleles that are donated are going to translate into something on the surface of a red blood cell. For example, let's call this our A blood sample right here. And let's imagine that A-blooded people, by receiving that allele for A blood, get these little triangles on the surface of their blood cells. Now, B-blooded people, we'll go ahead and make another column for that. Here's our little red blood cell. If they receive a capital B allele, they're going to have these structures on the outside of the cell, little half circles, little bumps sitting on the surface of their cell. These are called antigens. Antigens are little surface structures, sometimes made of carbohydrates, sometimes made of proteins, that allow bodies to recognize particular surfaces of cells and whether they belong in that particular body. Next, let's do AB blood. Let's draw a little red blood cell. And this one, we said both of the alleles would be expressed. So this one has the triangles and the little round half circles. So this one's surface looks a little bit different in the fact that it has both. Then we get to the O red blood cell. And an O red blood cell here has nothing on its outside. Now, how does that play a part in blood types? What's going on here? Well, here's the thing. Within the plasma of each blood type, sometimes you have what's known as antibodies against another antigen, and sometimes you don't. So for example, you have in an A-blooded person, you're going to have antibodies floating around that are called anti-B antibodies. And what they look like are little sticks with little tiny, almost like half circles on the ends of those sticks. These are made of protein. In a B-blooded person, they're also going to have antibodies, but they're going to have what's known as anti-A antibodies. And these will be little sticks, and they have these little wedge shapes on the end of them. There we go. On an AB-blooded person, they're not going to have any antibodies floating around in their plasma. So we can say no antibodies. There we go. And for an O-blooded person, they're going to have both anti-A and anti-B antibodies floating around their plasma. Now, what do these antibodies do? Well, they're going to be on the lookout for other blood types that may find their way into their body. And let me finish up my B here. So if I have anti-B antibodies and someone gives me a large quantity of B blood cells within my A body, what's going to happen are these little half circles on the end of these antibodies right here are going to start connecting with these little half circles on the surface of B red blood cells. And what's going to happen is they're going to lock them up and clump them up. So it's as if you have A blood, you're always on the lookout for B blood. And if it enters your body's going to clump it up, and it's going to go ahead and block blood vessels, which is very bad for you. Similarly, with the B-blooded person, notice they have these little wedge shapes on the end of their antibodies. So if a lot of type A red blood cells get in your bloodstream, what's going to happen are these little wedge shapes are going to connect onto these little triangles on an A blood cell, and they're going to start locking those up. So if you have B blood, you're always on the lookout for A blood getting inside of you. And if it does, your antibodies will clump it up. Now, if you have AB blood, you don't have any antibodies that go against A blood or B blood. So in this case, you just have those surface molecules that we called antigens. And then over here, if you have O blood, you have not only anti-A, but anti-B. So if someone was to put A red blood cells or B red blood cells in your body, you would clump up both of them. Let's take a bigger look for a moment at blood distribution in the United States and the world. Blood types all over our planet are different amongst our species. If you look at the United States, you can see that a great deal of the blood types that we find here in the US are O positive. However, almost equally high are A positive blood types as well. You can see that B takes up a smaller chunk of the population and AB even smaller than that. Now, notice you all see these pluses and minuses. We're going to talk about those in a little while, but there's a subset of each blood type called positive and negative, which can also lead to different transfusion issues. And again, we'll talk about that in a little bit. If we look at the world and its population, and we focus in on a singular blood type, you can see percent of the population that has the O blood type. There are some places where O completely dominates. 90 to 100% are all found in these blue areas. 80 to 90% are the dark red. So you can see a great deal of North America is either O or 90 to 100% or 80 to 90. 70 to 80% is kind of the lighter red, the pinkish color. But then as you drift eastward or westward, you're going to notice that those percentages change. You can do the same thing with the B allele here, where you can see the highest proportion of the B allele is going to be found in this central region right here of Asia, and we should say Eurasia. And then over here, we can see the A allele very low in some parts. South America, Central America, 0 to 5%. But that A allele is very high in other small regions, Southern Australia, Scandinavia, and then Northern Canada into Alaska. So the populations that carry blood types are always different. A long time ago, they were probably more stable, but as we know, the world is very small in a way that represents different people coming together and creating families and mixing blood types from all over the world. You can also take a look at different populations that you find all over the world. You can see some things that are fairly consistent in terms of numbers, and then you see some other examples where you see a lot of significant differences from other countries and other subsets of populations. So blood types can be studied and then linked to different things as well. So if someone is investigating something medically, they may see if blood type plays into it. Susceptibility of disease, predispositions for certain diseases to happen later in life. There's a lot of possibilities that they can look at and then study blood types as a potential influencer of that condition. The last thing we'll talk about is who are the universal donors and the universal receivers of blood and why. So if we take a look at this graphic right here, you can see our four blood types, A, AB, B, and O. And these four blood types are able to donate to specific groups and receive from specific groups. Let's take a look at A first. Notice that if you have A blood, you can donate blood to another A-blooded person. If you have O blood, notice you also have one of your rays extending to an A-blooded person. So the question comes up, why could an A-blooded person receive blood from an O-blooded person? Now, remember those red blood cells. Now, with the O-blooded look, the O-blooded phenotype of the blood cell itself, it didn't have A or B antigens on the surface. The surface looked kind of clean in terms of the A and B antigen. So with that, there's nothing to react with the antibodies, the anti-B antibodies floating around in a person's blood. So it's perfectly okay to give O blood to an A-blooded person. Next blood type, let's look at B for a second. B can donate to B, that makes sense because it's the same blood type. But notice again, O can also donate to B. And the reason for that, again, the O red blood cell is clean on its surface from A and B antigens, so it's not a problem donating it to B. Now you'll also notice that B can donate to AB, and also A, which I forgot to mention, can donate to AB. Now, why can A and B blood donate to AB? Well, if you remember what the AB blood looked like in the plasma, there were no anti-A or anti-B antibodies, which means that can then essentially receive blood from either A or B, not a problem. If we go right in the middle and hit AB blooded people, notice that they can donate to other AB blooded people. That makes sense. But again, A, AB, B, and O can all donate to AB blooded people because there's no anti-A or anti-B antibodies. And that makes AB blooded people the universal recipient of blood. Now let's look at our last blood type, O. Notice that it can donate to anyone, A, AB, B, or O. O is what we call the universal donor. Because of that lack of antigens on its surface, anti-A and anti-B, it can donate to any of the blood groups, so that makes it a universal donor. However, if you do have O blood, notice you can only receive blood from O. The reason why is because you have anti-A and anti-B antibodies floating around in your plasma. So if you were to receive blood from B, AB, or A, that blood would clump up inside of your blood vessels as those antibodies lock them up. If you want to try your hand at saving some patients by way of their blood types and transfusing blood into them, please go to www.nobelprize.org, find the educational games, and play the blood typing game. I think you'll get a lot out of it, and you can see if you understand the concept of who can receive what blood type and why. One last thing I'd like to discuss with you were those pluses and minuses that you saw from an earlier slide. Now, those pluses and minuses are also going to play into the ability to donate blood to another blood type. What are the pluses and minuses? Well, those are called your Rh factor. It's called a rhesus factor. This is another one of those antigens that sits on a cell's surface. This one happens to be a type of protein found on the outside of that red blood cell. You will inherit this or not inherit this based on your two parents. If you have this protein antigen on the outside of your blood cell, we are positive. We're Rh positive. If you do not inherit the protein, you are Rh negative. And that will also be part of your blood type as well. So we can subdivide blood types instead of the 4 going to 8 by way of this additional antigen on the surface. Now, in terms of the raw numbers, more people are positive than negative. Somewhere on the range of 80% are positive and 20% are negative. But it will definitely play into transfusing possibilities of who could give blood to whom. Now, notice right here, if we talk about AB blood, we said that AB blood was the universal recipient. And if you notice our top line, 1, 2, 3, 4, 5, 6, 7, 8. All 8 of the blood types that we can differentiate by terms of the ABO system and now the Rh factor system are 100% receivable by an AB positive person. Now, notice with an AB negative person that 4 of those have been X'd out. The reason why is if you are negative, you're going to have antibodies floating around your plasma that are looking for that positive antigen. And if they find that positive antigen, they can also cause clumping to happen within a vessel. So in this case, an AB negative person can only receive the 4 blood types, but in negative form. And if you did your nobelprize.org activity, you might have got a few wrong based on that idea. So we have to factor this additional antigen in when we talk about the ability to donate or not donate blood to someone successfully. Here we see an A positive person. They can receive O blood or they can receive A blood. But if we look at the A negative person, notice the O positive is taken out and so is the A positive. Again, that's because they're going to have anti-Rh factor antibodies floating around their plasma. Same thing with B. B can accept from O and B if you're B positive, but if you're B negative, you got to take 2 of those out. You can only receive from O negative or B negative. O positive people can only receive from O, and if you're O negative, you can only receive from O negative because you'll have the anti-Rh factor antibodies floating around, ready to clump up that blood. So we've reached the end of this lecture. Hopefully you're able to reflect back on your learning targets and see if those ideas are there and if you can explain them. So who was the individual that founded the 4 blood types? What are the 4 blood types? What is a gene? Where do we find it? What are homologous chromosomes? What is an allele? Can you differentiate between homozygous allele combinations and heterozygous allele combinations? Can you explain what being dominant means for an allele? Can you explain the special condition for codominance? And can you explain what recessive allele combinations do? Number 8, can you define a polygenic trait? And number 10, who is a universal blood donor and who is a universal blood recipient and why? I hope you learned a lot from today's lecture. Stay safe and I'll see you next time. Bye.